So we were doing about the translation process, and in the first video lecture we have done about up to the acceptor arm of tRNA structure. Now I will discuss about the cloverleaf model of tRNA, and then I will go through the how tRNA carries that amino acid into their acceptor arm with the help of that enzyme called amino acyl tRNA synthetase. Let us see. So let's begin. This is your second video lecture of your translation process. So I hope it will help you to in your examinations. Okay. So now about the cloverleaf model of tRNA. Well, the cloverleaf model of tRNA is a model that depicts the molecular structure of tRNA. Well, the structure of tRNA looks like a clover leaf, and according to the clover leaf model, the single chain of tRNA is folded onto itself to form 5 arms. So tRNA is normally a single stranded. Usually all RNA are single stranded, but there are some self complementary regions which leads to folding of the tRNA here, and that's why tRNA is folded onto itself to form 5 different arms. And as a result of this folding, the 3' end and the 5' end are near to each other. Now, the tRNA that we normally see is the 2D model. Okay, but if you see the cloverleaf model of tRNA, you will able to identify that the 3' and the 5' end located very close to each other. Whether you are seeing seeing the side view or through the normal cloverleaf model view. And due to its folding, as the tRNA are folding onto itself at certain regions, the 3' and the 5' are located very close to each other, and each arm that we are talking about, tRNA's arm, it consists of a basal stalk and a loop. The basal stalk is the place where complementary base pairing occurs, whereas the loop structure occur due to as the base pairing in that region does not occur. That's why when we see the structure of tRNA, we see some base pairing regions, which are the stalk regions, and we also see a loop like structure, the D loop, the anticodon loop, the variable loop, the T psi U loop. This type of loop like structure occurs due to non base pairing regions, and the stalk shows the base pairing regions. So the double stranded stalk shows typical base pairing, whereas the loops have does not have the base pairing. Okay, so the loop region are not involved in complementary base pairing, whereas the stalk region involved in the base pairing. If you see the picture, you can able to see. So you can see that these loop regions that I am talking about, the D loop, the anticodon loop, the T psi loop, the variable loop, these loops does not have complementary regions, and and these regions, you can see complementary base pairing has been occurred in these regions if you see it carefully. And this is the basal stalk, which shows typical base pairing. Loops does not involve in base pairing. Okay, and one of the arm which does not have a loop is called acceptor arm. So only one of the arm which does not have this loop, here no loop formation occurs, and that is called as acceptor arm. And this arm is actually the binding site for the amino acids. The amino acid will bind to this site. Amino acid will bind to this site of this acceptor arm with the help of the enzyme known as amino acyl tRNA synthetase. Okay, the second arm that you can see is the D arm. Okay, the second arm that you can see is the D arm. So this is the D loop, whereas this region is the D arm. Okay, so this region is the D arm, whereas this is the D loop. Similarly, this is the anticodon arm, and this is the anticodon loop. This is the T psi U arm. This is the T psi U loop. Okay, so and you can also see a variable arm as well. Most of the case we see a variable loop here, no arm. So the second arm is called the D arm. This has a synthetase site which recognize the amino acid activating enzyme. That means to say that this D arm that we are talking about, this D arm is the place where it is the um, it has a synthetase site. That means it will helps us to recognize the amino acid tRNA to identify that yes, this is the tRNA, and we have to catalyze the addition of amino acid to the tRNA's acceptor arm. Okay, so the D arm is also very important, whereas D loop is important in stabilization of the tRNA and also acts as a recognition site for the amino acid tRNA synthetase, right? Next is the uh, variable arm. Well, we cannot see the variable arm in this picture, but we can see a variable loop here. Well, variables arm may be present or may not be present. Now within the variable arm we can see only the loop, but no arm, no stem is present, no base pairing regions is being found here. Well, the T psi U arm, or you can simply say psi U arm. So where is the T psi U arm? This is the place where T psi U arm is present. Now this T psi U arm has a ribosome recognition site. This region has a ribosome. It has a ribosome recognition site. It has a ribosome recognition site. This T psi U arm. Okay, whereas the T psi U loop helps in stabilization of the tRNA and also for the attachment of ribosomes during translation process. So all these base pairing regions are actually the hydrogen bonds. So the cloverleaf model is fully stabilized by the hydrogen bonds, and this type of hydrogen bonding is called secondary hydrogen bond. Okay, so this was about the cloverleaf model of the tRNA. We can see the arm as well as the loops. Okay, here the arm has not been mentioned, only the acceptor arm has been mentioned here. This arm is the T psi U arm, this is the anticodon arm, and this is the D arm. These arms are actually are those regions where you can see base pairing has been done for stabilization of the tRNA. Okay, and it has also other roles as well. Like I have said, that D arm is the site where it recognizes the amino acid, which recognizes the amino acid activating enzyme. So amino acyl tRNA synthetase. Okay, amino acyl tRNA synthetase. This enzyme recognizes this D arm as well as the D loop and allows for amino acylation of the tRNA. That means it will helps to catalyze the binding of amino acid to the tRNA's acceptor arm. Well, we have also seen the T psi U arm. This T psi U arm has a ribosome bind at recognition site. That means this region will helps the ribosome to recognize the tRNA to bind to that site and carries out the translation process. Okay, so this was about the clover leaf model. The next that I want to talk you about, well, more things has been given. Okay. So this picture that has been shows, it shows the side view of this clover leaf model. You can see very clearly, you can see very clearly that how close is the 5 prime and the 3 prime regions, how close the 5 prime and 3 prime is located. Due to the folding of the tRNA, the 5 prime and the 3 prime are located very close to each other. Okay, if you see the side view, it will also shows you the same thing. Okay, well, we can also see the this is the ribbon structure of tRNA, the ribbon model of tRNA, which shows that this is the acceptor arm, whereas this is the 5 prime side, this is the T psi U loop, this is the D loop, which is present just behind the T psi U loop, the variable loop shown here, which shows in orange color. The orange color shows the variable loop. The D stem shows this one. The anticodon loop shows the an anticodon stem shows in the deep blue color, navy blue color, and the anticodon loop, which has the anticodon sequence, which will recognize the particular codon regions in the mRNA. Okay, specific for a particular amino acid, that codes for a particular amino acid. So this three-dimensional structure of tRNA has been shown here. Well, we draw the two-dimensional structure. This structure we normally draw. Three-dimensional structure shows a clarity of the loops of the stems that was hidden. Okay, so very clearly it shows the structure of the tRNA, that how tRNA binds to the mRNA codon regions with the help of this anticodon loop. Okay, and there it is, there it attaches the specific amino acid. Okay, so this is my structure that I have drawn. This is the structure of the tRNA. Now, the next thing that I want to say is about the enzyme which catalyze the attachment of amino acid to the tRNA, and the name of the enzyme is amino acyl tRNA synthetase. Now let us see what it says in these notes. So when a ribosome pairs with CGC tRNA with GCG codon, it expected to find an alanine carried by the tRNA. So directly it is saying that suppose during translation, the ribosome is carrying the translation process. Okay, and silent and during translation elongation, the ribosome somehow the ribosome identifies the CGC tRNA. Okay, sorry, again I'm repeating. Suppose the ribosome is translating a particular mRNA, and the ribosome encounter a region of the of the mRNA that has a GCG codon. The ribosome identify the mRNA having GCG codon during translation. So definitely the ribosome will allow a CGC tRNA to come and interact with the GCG codon. That means the CGC tRNA, that means the anticodon sequence of the tRNA will have CGC anticodon sequence that will base pair with the GCG codon region of the mRNA. And you know that GCG codon will code for an amino acid known as alanine, which will be carried by the tRNA. So each tRNA, if I say that how ribosome allows that particular amino acyl tRNA to bind to the particular codon region of the mRNA coding for that particular amino acid, it's because each tRNA is matched with its amino acid long before it reaches to the ribosome. In Bengali, if I say tRNA আগের থেকে স্পেসিফিক অ্যামাইনো অ্যাসিডের সঙ্গে বাইন্ড হয়ে থাকে, which has been catalyzed by the amino acyl tRNA synthetase রাইবোজোমে ঢোকার আগে. Okay, So long before translation process could begin, each tRNA binds with specific amino acid by the process called amino acylation and amino acylation. Okay, I think it says amino acylation. Yeah, amino acylation. And during this process, after this process, then when the elongation of translation occurs, the ribosome identify the codons of the mRNA, and the codon, suppose GCG codon, it identifies the GCG codon towards the 3' end, and for GCG codon it has to come a CGC anticodon tRNA, which will be carrying a specific amino acid called alanine. In this way, your tRNA is incorporated to the ribosome for elongation process. But how does tRNA attach with the amino acid long before the translation could begin? Now, the match is made possible by a collection of remarkable enzymes known as amino acyl tRNA synthetases. So, there are some specific enzymes called amino acyl tRNA synthetase, plural synthetases, which carries out the amino acylation of the tRNA, that is in simple attachment of amino acid to the tRNA. How does it do? How the enzyme do this process? These amino acyl tRNA synthetase, they charge each tRNA with proper amino acid. Charge means they catalyze the attachment of each tRNA with proper amino acid, thus allowing each tRNA to make proper translation from the genetic code of DNA into amino acid code of proteins. So, again I repeat, these amino acyl tRNA synthetases, they charge each tRNA, they make functional tRNA by attaching to a proper amino acid based on their anticodon sequence, based on the coding sequence of the mRNA also. These amino acyl tRNA charge each tRNA, or you can say activate each tRNA with proper amino acid and allow each tRNA to make proper translation, right? To make proper translation from the genetic code of DNA, or you can say from the genetic code of RNA into amino acid code of proteins. Now there are about 20 different amino acyl tRNA synthetase, and each amino acyl synthetase is specific for each type of amino acid. Now most cells have this number of amino acyl tRNA synthetase. These amino acyl tRNA synthetases are specific for a particular amino acid, and these 20 enzymes are widely different, each optimized for function with its own particular amino acid and the set of tRNA molecules appropriate to that amino acid. So each of these amino acyl tRNA synthetase are specific for each type of amino acid, and the 20 enzymes, these 20 different amino acyl tRNA synthetase, are widely different from one another, and each of these enzymes are optimized, or you can say they are effectively used for function with its own particular amino acid and the set of tRNA molecules appropriate to that amino acid. That means this, I can say that these amino acyl tRNA synthetases, each of them are dedicated for binding of a particular amino acid with respective tRNA molecules for particular codon or codons, because there are more than one codon which can code for the same amino acid. That's why there are 64 codons and 20 amino acids. So I hope you have understood what I have tried to say. I have said that there are 20 different amino acyl tRNA synthetase, and each of these amino acyl tRNA synthetase are different from each other, and they are dedicated for function with its own particular amino acid. So they are destined that they will allow each of this enzyme will be dedicated to bind that specific amino acid with the respective tRNA molecules for particular codons or codon, because there are multiple codon code for the same amino acid. I think you have understood what I have said. Now let us see. All amino acyl tRNA synthetase attach an amino acid to the tRNA in two enzymatic steps. So there are two steps. There are two enzymatic steps which causes all the amino acyl tRNA synthetases to attach an amino acid to the tRNA. Okay, so there are different type of amino acyl tRNA synthetases I have said, and each of these amino acyl tRNA synthetase are dedicated to attach specific amino acid to the tRNA. Now they do this attachment in two enzymatic steps. Let us see that steps. First is step number 1. In step number 1, it is saying that the step number 1 is called adenylation. Again I repeat, step number 1 is called adenylation. In adenylation step, the amino acid reacts with the ATP and become adenylated with the release of pyrophosphate. So what does it mean? What does it mean? In the step 1, it is saying the step 1 is known as adenylation. What happens in adenylation? The amino acid reacts with the ATP and become adenylated AMP with the release of pyrophosphate. Adenylated means your ATP, from the ATP, two phosphates are removed out and the remaining AMP attached to the amino acid. So it becomes amino acyl AMP. So in step 1, your amino acid reacts with the ATP molecule, and as a result, from the ATP, two phosphate molecules are removed out, so remaining AMP attach with the amino acid and form amino acyl AMP. This reaction is known as adenylation. I think you have understood. And the principal driving force for this adenylation reaction is the subsequent hydrolysis of the pyrophosphate by pyrophosphatase. So who is driving that energy for this reaction? The main principal driving force for this step 1 reaction, for this adenylation reaction, is the hydrolysis of the pyrophosphate. That pyrophosphate which was released from the ATP during adenylation, and this hydrolysis of the pyrophosphate done by pyrophosphatase. It is done by pyrophosphatase. So simply step 1 is called adenylation, where you see that amino acid reacts with the ATP, that causes release of two phosphate molecules from the ATP, becomes AMP, and the AMP now attach with the amino acid to become amino acyl AMP. This process is called adenylation, and the main principal driving force for this adenylation reaction is your hydrolysis of the pyrophosphate, and that is done by the enzyme called pyrophosphatase. Okay, always remember these words, that adenylation means transfer of AMP to to where? To the amino acid. Transfer of AMP to the amino acid, whereas adenylation simply means transfer of adenine. So this is a wrong word. If you say adenylation, you have to say adenylation. That means transfer of AMP to the amino acid to become adenylated. Okay. The next step that I will say is your tRNA charging. Okay, before that, before tRNA charging, we should do, let us see the picture of the first step reaction so that it will be easy for you to recite. Well, this is my diagram that I have drawn. This is the step 1, where you can see this is the amino acid. See the side chain, see the 4 functional groups that are present. So this amino acid reacts with a ATP molecule. It reacts with the ATP molecule. See the 3 phosphates which are located one by one in the ATP molecule. And in the first step, what I have said, that during the step 1 reaction known as adenylation, amino acid amino acid attach with ATP. Okay, it reacts with ATP with the release of phosphate and becomes adenylated amino acid. So you can see the amino acid now. This is the amino acid which you can see, it is now attached with the molecule of AMP, and the two phosphates of that ATP has been released in the form of pyrophosphate, as you can see in this diagram. Okay. Okay, so this is the first step, that we have to prepare adenylated amino acid. Okay, so you can see here. Okay, now the next reaction, the next step is called tRNA charging. The next step is called tRNA charging, in which the adenylated amino acid, which is your amino acid attached with AMP. Now this adenylated amino acid, or better to say adenylated amino acid, okay, the adenylated amino acid remains tightly bound to the synthetase enzyme that reacts with the tRNA. So the next step that we have said, I am saying, is about tRNA charging. In this step, the adenylated amino acid, which is your amino acid attached with the AMP, this amino acid which attach with the AMP now remains tightly bound to the synthetase enzyme. It remains tightly bound to the synthetase enzyme. That's the name, amino acyl tRNA synthetase enzyme. It bound and it reacts with the tRNA. And this reaction will result in transfer of amino acid to the 3' end of the tRNA. So the amino acyl tRNA synthetase attach this adenylated amino acid, and then it reacts with the tRNA. And due to this reaction, it causes transfer of the amino acid to the 3' of the tRNA by 2' or 3' hydroxyl and release of AMP. So during this reaction, what happened? What I have said, again I repeat, amino acyl AMP, which is your adenylated amino acid, that is your amino acid attached with AMP, now attached with the enzyme called amino acid tRNA synthetase. This amino acid tRNA synthetase then reacts with the tRNA and result in transfer of the amino acid to the 3' end of the tRNA via 2' or 3' hydroxyl and release of the AMP. So it means to say that if in the acceptor arm of the tRNA there is a sequence called 5' CCA 3'. So there is a sequence called 5' CCA 3' within the acceptor arm of tRNA. Now the adenine residue which is found on this 3' sequence, so the adenine residue of this sequence have to have its sugar molecule. Within this sugar molecule of adenine, amino acid will bind at the 2' or 3' hydroxyl group position. So here this reaction is catalyzed by your amino acid tRNA synthetase. I am repeat. You can see this CCA sequence. This CCA sequence is located on where? It is located on the 3' end, where I have also drawn. Look at this CCA sequence. This is called the acceptor arm, right? So within this acceptor arm, there is a CCA sequence. Okay, very good. Now in this CCA sequence, what we see in this CCA sequence, we see the adenine. In this CCA sequence, the adenine that we can see, in this adenine they have a sugar molecule also, right? They have a sugar molecule also because it is a nucleotide, so it has a sugar molecule. And within this sugar molecule, within this sugar molecule of adenine, your amino acid attach. Okay, and this attachment is done either on the 2' or on the 3' hydroxyl group of that sugar found in adenine. Okay, and this process is catalyzed by your enzyme called amino acyl tRNA synthetase. Okay, I think you have understood. Let us see the diagram so for your clarification. So you can see here what I have seen. So this is your adenylated amino acid. This is your what we can say. This is your amino acid, and this part is your AMP. Right now, this adenylated amino acid will actually attach with the amino acid tRNA synthetase very tightly, and then it will react with the tRNA. Mainly it reacts with where? Mainly it reacts at the acceptor arm region. On this arm of tRNA, amino acid tRNA synthetase will react. And due to reaction, what happened? What happened? We see due to this reaction, within this tRNA on the acceptor arm of the tRNA, we can see a adenine nucleotide on the acceptor arm. Now within this adenine nucleotide, the ribose sugar which is found on the adenine, which forms the backbone of the RNA, that ribose sugar, their 2' or 3' position is being used to attach that specific amino acid onto that region, and that is catalyzed by the amino acid tRNA synthetase. So here it is saying that thing they are saying here, it means to say that in the acceptor arm of tRNA, there is a sequence called 5' CCA 3'. Okay. Now the adenine residue that is found on this CCA sequence, they have its sugar molecule. Within this sugar molecule of adenine, amino acid will bind at the 2' position or 3' hydroxyl position, catalyzed by the enzyme amino acid tRNA synthetase. So in this way, your complete amino acylation is done. So in the first step, we can see that amino acid reacts with the ATP and form amino acyl AMP. That means amino acid attach with AMP molecules with the release of inorganic phosphates. And the main driving force for this reaction is the hydrolysis of that pyrophosphate by pyrophosphatase, which was released at that time. Second reaction is tRNA charging, where we can see that adenylated amino acid, that is the amino acid attached with AMP, they first remain tightly bound to the amino acyl tRNA synthetase. Okay, very good. After binding with the amino acyl tRNA synthetase, now this enzyme will react with the tRNA, and during the reaction it causes transfer of the amino acid either to the 3 prime end of they cause attachment of the amino acid to the 3 prime end of the tRNA, that is on the acceptor arm. This amino acyl tRNA synthetase catalyze the binding of the amino acid to the tRNA via, that means through, 2 prime or 3 prime hydroxyl and release of the AMP. Now, where do they find the 2 prime 3 prime hydroxyl? Within the tRNA, we can see a acceptor arm, and within the acceptor arm, there is a sequence called 5 prime CCA 3 prime. The adenine residue of that CCA sequence is involved in attachment of amino acid to the tRNA. ठीक है, catalyzed by the amino acyl tRNA synthetase. Okay, more specifically, within the adenine residue, there is a sugar molecule, the ribose sugar, and within the sugar molecule of adenine, the amino acid attached to the 2 prime or 3 prime position. So this is the whole reaction that I have mentioned with you. Okay, I have mentioned with you. So just see through the steps that I have said. And this is the reaction that you can see. This is the book that I have followed here. Well, same diagram I have drawn in the previous pages that you can see. Amino acid, you can see amino acid attached with they react with the ATP and release of pyrophosphate, and it forms adenylated amino acid now. That means the amino acid now attached with the it now attached with the AMP molecule. So if this is the amino acid, this is your AMP molecule. Okay, with the release of pyrophosphate, this pyrophosphate will be further hydrolyzed by pyrophosphatase, which will stimulate that reaction to be done. Second reaction, we can see that this amino acid, this adenylated amino acid that you can see, they are now attached with the enzyme called amino acyl tRNA synthetase, which has not been shown in the diagram. And attached, after attachment with the amino acyl tRNA synthetase, now they are trying to react with the tRNA in the acceptor arm, and within the acceptor arm there is a CCA region. You can see the CCA region. Within the CCA region, you can see an adenine region. Now within the adenine region there is a sugar molecule also, and that sugar molecule, it catalyzes the binding of the amino acid to the tRNA. So you can see how amino acid has been attached to the tRNA molecule with the release of the AMP. Okay, so this is a high energy bond, which is actually an ester linkage. I have said before that that linkage is a ester linkage formed in the formed in the sugar molecule of the adenine of the acceptor arm. Okay, so that ester linkage is highly energetic bond, and AMP is get released. Okay, so up to this much I have said my lecture. in the next lecture, I will more say about the amino acid tRNA synthetase, their function. Okay, so this is your second lecture. I hope through couple of series of lectures I will finish my whole translation process. So if you have some doubts regarding this part that I have mentioned to you just now, you can ask me in the WhatsApp group. Okay, so just I am showing you the last diagram of this. In the notes you can also able to see. So this is the first step of amino acylation of tRNA, and this is your second step. Okay, so that's all for today's lecture. After of some period of time I will after some time I will discuss about the more about the amino acid tRNA synthetase with the other components of the translation machinery like ribosome, and we will begin our translation process.